So today is Wednesday, 8th of April, and this is the first of the final 4 lectures in this class, because on the schedule I had a lecture on April 22nd, and that turns out that's a no class day. So we're now in the home stretch of lectures for this. This one I'm going to talk about today, and driving gradients for movement in the phloem, and then converting that step by step to harvest index and carbon partitioning, because all that movement to fruits has to be in the phloem. This part here is very well covered in every plant physiology textbook. How the water potential movements cause flow in the phloem. So I refer you to the chapter on phloem transport in any plant physiology textbook for the basics of this. This one happens to be Thais and Zeiger, a more recent one, but these principles haven't changed for several decades. Let's summarize the cover of the book, and we now have covered every topic in here. The final one is movement of water from the soil matrix up to the plant, where we have solute potentials and turgor and total water potential. And you'll see we got every single equation now has been covered in here, so you know we're getting close to the end. This is the syllabus right there, and down here at the end you can see that we're now April 8th, assimilate partitioning. I'm going to talk about root systems and root absorption. That's another uniquely whole plant aspect of this, and I'm going to especially talk about freezing stress in these final lectures. There's 5 lectures on here, and I'm going to collapse them into 4 lectures to get it to the end of the semester. So let's review terminology, because some of these subscripts are not universally used. I mentioned this in the last lecture, and as a student this can just drive you crazy when people don't always use the same symbols. But hey, the whole world doesn't all speak the same language either, so you just gotta, the only solution is to be bilingual. Here's the written out, here's the terms. This solute potential, which is by things in solution, is also called psi with the Greek letter pi. So when you see this, it's exactly the same thing. I haven't studied why some people use psi s and psi pi, but this also means solute potential. We already talked about this as being turgor and pressure, but most commonly this is P for pressure. Now over here, psi W, often people write this with no subscript at all. It's just psi, and that always means total water potential. Matrix, fortunately, that's all I've ever seen for that is psi M. So that's in gravitational, we ignore that. I see people, sometimes I see people writing psi G for gravitational potential, or psi Z, but this is a minor component. This M is only in soils. There's no matrix potential in plants, but this is by far the dominant ones in soil. And by the way, there's no turgor potential in soil either. If there was, we'd have maybe the Yellowstone Old Faithful, maybe that's turgor potential in soils blowing up, but that's pretty rare. But water would be coming out of the soil if there was turgor pushing up. And so as soon as we get into plants, this goes away and these two dominate. And today we're going to focus on the ratios of these two, because that's key to understanding driving gradients in the phloem. So Let me attempt to draw a picture and fill it in. It's going to be big right here in the middle. Here's Xylem. And over here in green is Phloem. And I drew these extra big because this is, you really need an understanding of plant water potential to understand this Phloem. Now, why did I draw this one green and this one blue? No, it wasn't random. Water moves in the xylem and syrup moves in the phloem. This is very dilute and high velocity, so if I put some numbers on here, this is a whole bunch of arrows going up. Huge velocity of water going up. The phloem, on the other hand, is on the order of 1/100th the flow rate of the xylem. So here we're going to have little tiny arrows, just a couple little arrows pushing down in the phloem. But they're in opposite directions. How do we do that? And by the way, I drew phloem in green because if we do cell walls in the xylem, these are just little cell walls with a big open space. The xylem is not living tissue. This is a plastic soda straw sucking up water. The phloem has, as you will read or might have learned in some other class, it's living, and these are not open cells. These are what's called sieve plates. I won't write that on here, but you can look it up. So the phloem has a bunch of semi-permeable membranes for flow, whereas this is just an open conduit. So how does water go up one and come down the other when water always flows downhill? So let's put some numbers on here. Let's say, in this example, now I'm going to attempt to use the same numbers in my drawing here as are on a handout that I'm going to send you. And I could put the handout up there, but I've got them written here so I can put the same thing. But if I go through them step by step, I think it'll be more clear than just showing you a handout. First of all, now we're going to start with brown because we've got psi matrix down here, and we're going to call that 0.4. Everything in this thing is all megapascals. That's the universal unit. And the numbers are all 1, 2, 3. They're integers with tenths. Now, matrix potential 0.4. Already I've made an error on the board. Everything is negative numbers. That's just a fine number, but it's all negative. Negative, negative, negative. Except for one. We're coming to the one that's positive. But this is typical. Remember, field capacity, FC, is minus 0.03. And PWP, permanent wilting point, is minus 1.5. Suddenly, these numbers that you memorize for beginning soil science become important. And look, minus 0.4, that's typical. It's moist soil, and there's not water stress. It's in between these numbers. That's our starting point. We can run these analyses when the soil's really dry, but let's start with moderate. Water flows downhill. So now it's going into the cells of the root. Let's see, let me give myself some space right here to write some numbers. It's going in here. So if now inside the plant, this, by the way, this is psi water potential, psi W right here, total, which is equal to matrix. We're not assuming any salinity in this. It's just water down to soil. Water flows downhill, so this inside the plant has to be a more negative number for water to flow into the roots. And in this example, we're using minus 0.6. This is more negative, water moves in here. Here's the key. Solute potential, how did this get more negative? It's solute potential, change my number here, or my arrows, is minus 0.1. So there's some salts in here. Not many. This is pretty dilute solution. But wait a minute. Didn't I just tell you that psi total has to equal psi s plus psi p? What happens to psi p? Look, psi p for pressure minus 0.5. The water is sucked into here, and it's a negative potential. Minus 0.5 plus minus 0.1 equals, down here we got minus 0.1 plus 0.5 minus 0.6. There's the math. So the water's moving into the xylem. That's the first step, and it's sucked in because it's sucked up here on the top. The leaves are sucking it up through this plastic soda straw. So now we have to have more suction up here to get the water to move up. So up here we have psi, and I'll put W again for water, minus 0.8. So more negative, water's going up. I got to do some more erasing to give myself room. Psi solute potential up here, minus 0.1. Still very dilute, not many ions in this, it's just water. And you can see from the math, the pressure or Turgor potential now, minus 0.7. Minus, I don't have to put all these equal signs, I'll just put a colon. And there we go, so this plus this, minus 0.8. So now this water's sucked up, it's all good, water's moving to the top of the plant. If water flows downhill, is this some kind of perpetual motion machine that it's somehow magically coming the opposite direction? Here's what happens. Here is a leaf. It's not drawn to scale because I'm out of room. But here's a leaf right here. What's that leaf making? Sucrose. A lot of sucrose. A concentrated solution of sucrose, and that's going right into the phloem. So now we have a lot of sucrose right here. And this syrupy thick solution sucks water into it. And if we look at the water potential up here, we'll put the sucrose up here. Psi W is equals minus 1.1. Big deal. This is more negative than this, and so water's going to move from the xylem to the phloem because it's following the water potential gradient. Psi solutes, ooh, they're through the roof. There is so much sugar in there, it's a really negative osmotic solution. This, minus 1.7. All of a sudden we went from a dilute solution to a concentrated solution. And now we do the math. What's the pressure potential right here? It's plus 0.6. For the first time, there's a positive number here in the phloem, a positive pressure potential. Here it's negative, negative, but all that sugar sucks the water over, and this has turgor in the phloem. Remember, it's living. Here's the dotted line. It's living. Down here, look what's this at the bottom of the plant. Now, this could be, to be correct, this could be, here, I'm going to show you this in red. This could be a tomato fruit. It could be some sink, something that's using the carbohydrate. Of course, it can also be a root tip down here, growing into the soil. But someplace on the plant, if it's a tomato plant, it might be going up, but you get the idea. Someplace on the plant that's using all of this sugar that's concentrated up here, and it's moving down here. Guess what? The sugar moves out of the phloem here into the sinks. So all of a sudden, this is more dilute down here because the sugar's moving out. One end of this is loading it in, the other end of it's sucking out. Where's my green marker? And down here, we get a water potential of minus 0.4. Wait a minute, didn't you just tell us that water flows downhill? How can it go from a minus 1 to a 0.4? This is not more negative. This does not follow the rules. So how can this system work if it's not more negative? Look what happens down here. The solute potential, colon, get my numbers right, is minus 0.7. So look what happened to all the sugar. It's minus 1.7 here, a lot of sugar, it's moving out, so now this is more dilute. And now we do the math. Psi P down here has to be plus 0.3 to make the math add up because of this right here. So look what happens. It's plus 0.6 here, it's plus 0.3. This is a more positive number than this. So this is what we call the pressure flow hypothesis. In living tissue, in the phloem, turgor pressure is what's pushing the solution around. In dilute water, in non-living tissue, it's water potential, which flows from the soil with water potential. But here, it's a buildup of sugars, and those sugars suck water in because of super negative osmotic potential. And the turgor up here pushes it down. So it's a little like, let's go back to our beaker here. Here's our semi-permeable membrane. If we put lots of sugar over here, I'm going to put sugar, sucrose. Sucrose is the dominant sugar. Before we had salt in here. Now we put sugar in here. This is what's going to happen. It's going to move over here. Guess what? If we have a little tube right here, it's going to flow in the phloem back over on this side. So this little diagram right here shows water in non-living tissue going downhill, and yet there's a positive turgor pressure that can push it back the other way. That's how this works. Now, the reason this is a big deal is what happens if we do a matrix potential. Just let's take it to -1. It's not even permanent wilting point, but suddenly this soil dried out. I won't go through specific numbers, but when this dries out, oh wow, the plant has to suck harder to get water, for sure, and the phloem movement is significantly reduced when the soil gets dry. It's going okay as long as the soil is wet, but there's less of a gradient here to move water in and less positive pressure. So one of the consequences of drier soil is reduced phloem transport. And we think, okay, the leaves won't be able to cool as much, but reduced phloem transport is a big effect of drier soils. This diagram, every plant physiology book has some version of this in some way. This diagram here is, I can just hold this up, but I'll send you this electronically. This is a diagram exactly like I've drawn on the board. It just shows all the numbers and some explanations of this, but it's from Thais and Zeiger, the most recent, the 6th edition, and it shows how the water moves through the whole thing. Over the decades, teachers have gotten better at explaining this, but this is also called the Munk hypothesis, a German guy that first proposed it, and it's pretty well established this is the mechanism of phloem transport. But sometimes we still call it a hypothesis because other people have proposed different ways this could happen. So, but you can see how you got to have a good understanding of this and this and this to understand water movement in the plant. This causes water movement in the xylem. Here, let me put it up here. Psi W xylem movement. Psi P is, this is for pressure. But it's easy to remember. PHLO, phloem movement. It's also what drives movement in the phloem. So it's helpful to have a sense of how many ions it takes to cause this kind of movement in the plant, because a -1 water potential, it doesn't have a lot of intuitive meaning, but fortunately there's an equation to go from the weight or the amount of salt to water potential. And it's a pretty simple equation. And where am I going to put this? Over here. This probably is, let me see, purple. It's a different thing. Solute potential, or psi pi, either one, equals, now first I'm going to put the fish symbol. It's proportional to concentration of salt. Very intuitive. But there's got to be more to it than this. This concentration is in moles per kilogram of solution. Now, remember how we talked, wait a minute, moles per kilogram of solution. Why didn't you just write moles per liter of solution? This is driven by the weight, the ratio of moles per unit weight. Remember from beginning chemistry, molar solution is moles per liter. It's amount of salt per unit volume. A molal solution is moles per kilogram. And you say, well, why do we have two terms? A liter is a kilogram. Aren't we just splitting hairs? When you get to constant, in dilute solutions, these are the same thing. And dilute solutions are the case in most of biology, except right in here when we really have a lot of salts. I could have done a demo of this, but if we have a liter of water, here's a liter. You gotta do a little imagination. Take the top off. If I put a teaspoon of salt in there, what happens to the weight? It gets bigger. You have the weight of the salt and the weight of the water. What happens to the volume? Nothing. Goes into solution. The volume doesn't change. But you can see how as you put more in, the volume can stay constant and the weight can increase. And there's certain salts, we could do it, the calcium nitrate is a great one. You start to put that in and the weight goes up and the volume goes up. But that's why we have two terms. This concentration, I'll leave this right here, is in moles per kilogram. All right, how do we turn this proportional into an equal sign? So if we do equals, we bring back the ideal gas law constant, R times R. And of course, we have to bring back Professor Kelvin, temperature in Kelvin. And this is getting close to equal, but there's one more term. And this is shown as a lowercase I. This is an ionization constant. And if we have, I'm going to need to erase some stuff to get more room. If we have, here, you write it this way. The ionization constant for sucrose is 1. It's not really an ion, but it's one molecule. If we do sodium chloride, Cl minus, there's two ions, one sodium, one chloride. It's 2 for a salt. If we do now, you can imagine calcium chloride. There's 2 minuses, and this one's 2 plus. It's 3. So it depends on the salt and how many ions it breaks into. But as a general rule, sodium chloride, the dominant salt that drives osmotic potential in soils and saline soils, is 2, and sucrose is 1. And then we take the concentration times R and T. So if we put some numbers in here, this, now I'm going to change colors. R, with the units that make this cancel, 0.0083. Let me make sure I got my numbers right. Yes, 0.0083. Now we take Kelvin temperature and we do 273 Kelvin. Let's not do that. Let's do 303 Kelvin. This is slightly warm. And it's like, what is that? 270, 28. It's in the 30s. It'd be a warm day, but it's still pretty close. These two numbers together, oh, guess what? I made a math error already. This is minus. Always has to be minus in here. So we take these two together, and it comes out to be 2.5. 2.5 something. Now, if this was 1 and this was 1, a 1 mole solution, 1 molal solution would be minus 2.5 megapascals. So always in megapascals. So at a slightly warm temperature, 1 mole of solution is minus 2.5. Wow, that's already below our permanent wilting point in soil. This is a pretty negative suction, which is why we have big problems with saline soils. But let's work through this. If it takes 1 mole, and this is 1, sucrose, I've worked this out here, is 342 grams per mole. Big molecule. And a kilogram, this would be 342 grams per kilogram. And I'm going to put liter because it's almost the same. But this is 34%. This is a very sugary solution. This would be some fraction of a cup of sugar in a liter of water. This is a real concentrated solution. If this is sodium chloride, sodium is 23 grams per mole. Chloride is 35 grams per mole. You can look them up in the psychrometric chart. This is 58 grams per mole. So this is 58 grams G per kilogram. But now look, the I is 2. So we get to divide this by 2 for the ionization constant, and this comes out to be 29 grams per kilogram. Look at this. Where can I write this? 2.9%. The point here is that ions have a big effect on osmotic potential, and sugars, you really need a lot of, a heavy weight of sugar for ion potential. I should do this as a demo. If we put in enough salt to make seawater, we would get minus 2.4, and no one would drink it. It's like, whoa, way too salty. If we put in this much sugar, it would be very, very sweet indeed, but you could still drink it. It's still, it's just sugar. So it gives you a sense of the osmotic potentials and the weights. And these things all come from this equation right here. And this, I don't have to show it up there. This is also in plant physiology textbook, how to convert moles of solute into water potential. And I've worked out some of these examples on this sheet to show you the potential. Okay. How does this relate? Do you guys know when we started? 10 after? Okay, good. How does this relate to the forms of carbohydrates? Well, a lot of sugar right here. But what about all that sugar? Isn't it built up down here? And don't we get a problem with there's so much sugar it can't keep sucking water? Here's why we don't have a problem. It's basically back pressure. Here's why we don't have a problem. What happens to sugar? We're going to go here, and here, and here. The sugar in the sinks, what form of carbohydrates are in seeds, like rice and corn and wheat? Is there sugar in those? No. Starch. And starch is a bunch of sucrose molecules all linked together. But guess what? Starch is not only more stable, but it is not water soluble. And you can show this even in a kitchen. If I have starch in here, we're going to do a...
There's starch right there.
Oh, is it in the shelf? Or?
Yeah.
There it is. Perfect. Yeah. Here. We're going to take our soil plant atmosphere continuum and sacrifice this to science. This is 13 grams of salt, and 13 grams of salt is slightly less than a tablespoon. So if we're relating plant water potential to what you do every day in a kitchen, a tablespoon of salt in a liter of water, meaning a quart like this, is -1 megapascal. I'm going to put that up here. We'll put this right here. 1 TBSP of, I'm going to put salt, because, and now the sodium chloride is the salt, equal, per liter is -1 megapascal. You can do the math yourself. It's 13 grams. A tablespoon is technically 15 grams, but we're pretty close. So it gives you a sense. If you put this much salt in here, could you drink it? Yeah, you could. It wouldn't taste great because it's pretty salty, but it doesn't take much. Now I should have worked out sugar, because 342 grams of sugar, there's 250 grams of sugar in a cup. So this is 1.5 cups of sugar. Wow. You know, as you're doing canning, you put wow, that would be a lot of sugar. Now let's put in starch. And here's a spoon. Here's the water, here's the starch. You can do this at home, but if I do it, it's more memorable. Put it in. Put it in, put some more in, put a bunch in, stir it. Does that go optically clear? No, it does not. And we got a little extra starch here. It's cloudy. If it's cloudy, that means it's in suspension, not solution. Doesn't matter how long I stir this, it's not going to ever get clear. Now we're going to do an experiment. 2 days from now, we'll have the next lecture. I'm going to leave this jar right here. What's going to happen? The starch is all going to be in the bottom, and the top will be optically clear because it's settled. It'll take 8 to 24 hours for it all to settle. The starch is not in solution, so guess what? If it's not in solution, it does not affect water potential. And that means this whole system can keep going as long as the plant's storing it as starch or something else. It can be proteins, it can be cellulose, everything else, but the dominant thing is starch, a lot of starch. And there's no effect of starch on water potential. It has to be in solution to affect water potential. So that's what keeps this going. Photosynthesis with sugars, they're in solution, they flow, and then they become starch down here, and it keeps going. So what happens during the day to these forms of sugars? And that's this graph right here. Let me see. I'm going to blow this up. Big. Refocus. Remember our carbon use efficiency graphs, photosynthesis in the day, respiration at night. This was a 12-hour day of light. Light comes on, but look what happens. This was in constant light. It was electric lights, so the graph was steady. Photosynthesis started out high in the morning. Ooh. Went down during the day, and the same thing happens every day. What's going on? People more productive in the morning? Some people are. But this was constant light. With constant light, this went down because of what we call feedback inhibition. It's feedback inhibition of photosynthesis. The system gets clogged up. This freeway down here, it can't move sugars fast enough. It gets clogged up here, they get clogged up in the leaf, boom, and photosynthesis slows down. Feedback inhibition is a real bad thing because here we are, everything's working, the leaf can't get rid of the carbohydrates. Because this is universal, we see it, and especially bad if it's in sunlight, because now you have a big curve in the middle of the day where there's really bright sunshine. All that sugar has to go somewhere, and it all has to follow the rules of driving gradients. So when we do an analysis of this, we get, let me give myself a little room right here and erase that. If this is the leaf up here, and this is the phloem, and here's the root right here. This is called the source, and this is called the sink. Draw a picture of a leaf. And the source is virtually always leaves, and this is called the sink, which is where it goes, and this is a nice diagram. A couple things have to happen. This is not a direct plumbing connection right there. It has to go across the membrane. And this sucrose has to get loaded into the phloem. So all this sucrose, it has to be dilute, and it gets loaded in here, pumped. It's an uphill gradient. Here's just a few sugar molecules in the leaf. It's pumped into the leaf using ATP, pump, pump, pump. This is super concentrated, drives a gradient. Down here, this is also not a direct plumbing link. So this is also goes downhill, and this is downhill. So this could happen passively, but this most definitely does not happen passively. It's called Phloem Loading, and it's ATP pumping it into the phloem, and it takes a lot of energy to do this. So as we have studied this in the quest to get higher and higher yields, the first place that might be limiting is Phloem Loading. L-O. And then down here is unloading, L-O-A-D. And in between, how about the freeway between them? Maybe this tube isn't big enough, and it's restrictive. And this is number 2, it's just transport. So to make an analogy, this is a great analogy to getting from here to Salt Lake City, where the on-ramp is really crowded and you got to buy a ticket and pay cash to get loaded, and it's hard work to get on the freeway. But man, once you get on that freeway, smooth sailing. All of our studies indicate that transport is not limiting. Once it's in the phloem, it moves. So we're looking at how to make phloem loading better and how to make unloading better down here. But these are the 3 sources of this. So I got a few minutes left, and this is helpful to link this now to what happens right in a leaf. We know that if it goes to starch, we're good. Now, way over here, a great big leaf. We're going to look at this. Now this is a leaf, but it's also a single cell. Here's a cell. So we have sun coming in. I'm going to put C and a circle for a chloroplast. We got to show sun coming in. Here. Okay. Now, this makes sugar. So sucrose. And if this was all it was, the sucrose can get pumped out, right out of the leaf. And if this was the whole story, it'd be pretty simple. But over here, shown in black, the vacuole of a cell, sucrose can get stored as starch in the vacuole. And that's not mobile, it's not, but it gets it out of the water potential stream, so all day long, VAC, vacuole, all day long starch builds up, because it's rush hour. And people just park their cars and wait, and guess what? Then night comes, back to sucrose, and back out. So this buildup of starch really helps the plant keep going, because it can make starch right there in the leaf. Now it turns out plants put starch right in the vacuole, or in the chloroplast. Wow. Some plants store a lot of starch right in the chloroplast, and there's electron microscopes of chloroplasts just bulging out because they're so full of starch. And for a long time we thought this can't be good, it's so packed in. But now we know it didn't matter. I mean, they can store it in the chloroplast and it can keep working as long as it isn't affecting the water potential. But you can't move starch, not soluble. It just stores it day and night. So that takes us to let me just draw this. I think it's better if I draw it. Put it in here. If now we make a graph, I can erase that. Here's day, and here's night. And this is carbohydrates, carbos. Day, these build up. This is right here in the leaf. Night comes, they go down. So up and down. But this is both forms of carbohydrate. Some of this is sucrose, some of it's starch. And we spent as scientists spent a lot of time analyzing where the starch goes because we're trying to get more movement, more photosynthesis, bigger fruits, higher yields. Is this part limiting? Yeah, we think it is. We did giant pumpkin analysis for record yields, incredible sized pumpkins. So some years ago, some scientists got involved and they said, do you think this phloem could be limiting for a giant pumpkin? There's an awful lot of transport to that pumpkin. And look what they found. This is a paper, I'm going to show you, can we see this right here? Probably. This is a paper in one of our more prestigious journals, Plant, Cell, and Environment. Here's the title. This is just a couple of years ago. The Making of Giant Pumpkins, How Selective Breeding Changed the Phloem of Cucurbita maxima, is the Latin for the pumpkins, from source to sink. They found that the breeders had been selecting for bigger and bigger phloem, and that's how the pumpkin got so big. And they analyzed small pumpkins, and as they got bigger, the diameter of the tube got bigger. So can we do that with other crop plants? Well, not only can we, but we almost certainly have to maximize this system to get more to the sink of the plants. One more thing. This is a handout, and this one is small, so we're going to put it up here. At least I think we can get it bigger. That's big enough. This looks, I don't know, like some sort of an MC Escher diagram. This is a disc of a leaf, and we fed this leaf. We didn't do it in this lab, but we fed it radio-labeled carbon dioxide, which we always show here. If we have radio label is 14 CO2, so that 14 carbon becomes 14 sucrose, and it becomes 14 starch. This is the value of radio labeling things. The carbon molecule, we can trace it where it goes. So photosynthesis went into sugar, and then a few hours later, we took, and then you can do a radiograph where the radio label compounds give off energy. This is all the veins of phloem, or the phloem tubes in a leaf disc, because the sugar was loaded in there. So the 14 label was concentrated in the phloem, and it's a beautiful example of phloem loading. A few hours after this labeling, it was all packed into the phloem, ready, sitting there in line, waiting for export to the rest of the leaf. This diagram is a simple diagram of the gradients, sugar concentration, distance from source. So right here is a leaf, and boom, it gets loaded into the phloem, so it's packed in, high concentration, and then it comes out of the phloem along the way, and finally it gets to the sink, and it goes back downhill. So this is a nice diagram of the energy involved. The concentration goes up and then back down. This is from another part of the plant physiology book, concentration of xylem and phloem. This is really instructive. Sugar in the xylem? None. This is just water. Amino acids are really mobile, so there's amino acids, and the rest of this is nutrients in the xylem. They're being moved around, drawn up for the roots. Now we go to the phloem, boom, the sucrose is huge. Amino acids are huge. These are all very mobile in the plant. And these other compounds are also mobile in the plant. But keep in mind, this is very dilute, but the flow is maybe 100 times faster than this slow, concentrated flow. Then finally, the fourth and final picture on here is just a leaf. I'm so zoomed in, it's hard to see. Let's take the top part. This is a young leaf. Right now, this time of year, the buds are coming out. Here's photosynthesis unfolding. Photosynthesis peaks and it goes down as it gets older. Respiration goes down. And at the same time, we have sucrose in the leaf. Sucrose builds up because of a lot of photosynthesis and then goes back down. Phosphorus is plant nutrition, but it has to import phosphorus when it's young, and then as the leaf gets old, it can export phosphorus. But the key here is the big buildup of sugar as that plant gets going. Let me close on this. This is a diagram that I sent last week. I don't know that I talked about it in class. Study this, make sure you understand it. This is just our simple beaker of water with a cell floating in it. and what happens to the water potential. I think if you look at this and look at the numbers, you'll say, okay, it starts out all zeros, then we put some sugar in there and we get some positive numbers, and then it wilts. A flaccid cell is when the turgor pressure goes to 0. What happens? And then turgid cell expansion. So take a look at this diagram in the handouts and make sure you understand it. This is a good place to stop. We will finish this up next time with a little more analysis of harvest index of the plants and how all of this translates to harvest index. And then we'll move into a few miscellaneous topics: root systems, how does water potential of a plant affect freezing? That's another lecture, but the couple of the lectures are still based on plant water potential. We'll keep studying. We'll see you next time.